These studies, what? These studies show that the cell membrane is mainly composed of lipids and protein. हाँ, जो स्टडी किया साइंटिस्ट उससे पता चला कि सेल मेम्ब्रेन जो मेनली कंपोज होता है लिपिड्स एंड प्रोटीन से. द मेजर लिपिड आर फास्फोलिपिड दैट आर अरेंज इन अ बाइलेयर. हाँ, पता चला कि मेजर लिपिड जो होता है वो फास्फोलिपिड है. प्लाज्मा मेम्ब्रेन में जो मेजर लिपिड है वो फास्फोलिपिड दैट आर अरेंज इन बाइलेयर. कैसा? बाइलेयर में अरेंज होता है. ओके. Also, the lipid are arranged within the membrane with the polar head toward the outer side and the hydrophobic tail toward the inner part. हाँ, important है. Also, the lipids are arranged within the membrane with the polar head toward the outer side. Polar head माथा जो होता है outer side होता है and the hydrophobic मतलब water repelling tail toward the inner part. Okay. This ensures that the non-polar tail of saturated hydrocarbon is protected from the aqueous environment. हाँ, assertion region यहाँ बनेगा. Okay. Ensure that the non-polar tail of the saturated, कैसा? Saturated, हाँ, hydrocarbon is protected from the aqueous environment. In addition to the phospholipid membrane also contain cholesterol. हाँ, in addition to the phospholipid membrane also contain करता क्या? Cholesterol. Laterally, biological investigation clearly revealed that the cell membrane is also possess protein and carbohydrate. कौन? biological investigation clearly revealed that the cell membrane also possess protein and carbohydrate. कौन? biological investigation. यहाँ पे important है क्या? biological investigation. okay. The ratio of the protein and lipid vary constantly in different cell type. ठीक है. The ratio of the protein and lipid क्या कहता है? Vary constantly in different cell type. In human cell, the membrane of the eukaryotic cell have approximately 52% protein and 40% lipid, depending on the S of excretion. कैसा? S of excretion. S of excretion, membrane protein can be classified into the integral and peripheral. Peripheral मतलब बाहर, integral मतलब अंदर. Peripheral protein lie on the surface of the membrane while integral protein are partially or totally buried into the membrane. Okay. देखो ये क्या है? हेड है. हेड शुगर है ना, पेरिफेरल प्रोटीन बाहर वाला. ये क्या? फास्फोलिपिड जो बाइलियर में अरेंज होता है. ओके. ये क्या? इंटीग्रल प्रोटीन जो अंदर में प्रेजेंट होता. ये क्या है? कोलेस्ट्रॉल. ये क्या है? कोलेस्ट्रॉल पीला पीला. Fluid Mosky model of plasma membrane. As improved model of the structure of cell membrane was proposed by the Singer and Nicholson in 1972. Important data. Improved model and the structure of cell membrane was proposed by Singer and Nicholson 1972. Widely accepted as a fluid Mosky model. According to this, aqueous fluid quasic fluid nature of the lipid enable the lateral movement of protein within overall bilayer. This ability to move within the membrane is measure its fluidity. According to this quasic fluid nature of the lipid enable lateral movement. कैसा मेथड? Lateral movement of the protein within the overall bilayer. Okay. The ability to move within the membrane is measured as its fluidity. The ability to move within the membrane is measured as its fluidity. Okay. The fluid nature of the membrane is also important from the point of view of the function like cell growth, formation of intracellular junction and secretion and endocytosis and cell division. The fluid nature of the membrane जो है ना also important from the point of view of the function like cell growth, formation of intracellular junction and secretion and endocytosis and cell division. Okay. One of the most important function of the plasma membrane is the transport of molecule across it. One of the most important function of the plasma membrane is the transport of molecules across it. Yes, the membrane is selectively permeable to the some molecules present on either side of it. ठीक है, द मेम्ब्रेन इज सेलेक्टिवली प्रेमेबल होता है टू सम ऑफ द मॉलिक्यूल्स प्रेजेंट ऑन अदर साइड. Many molecules can move briefly across the membrane without any requirement of energy, and this is called passive transport. Neutral solute may move across the membrane by the process of simple diffusion along the concentration gradient from higher concentration to lower water. Water may also move across the membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. Movement of water by diffusion is called osmosis, as the polar molecules cannot pass through the non-polar lipid bilayer. Polar molecules जो होता है cannot pass through the non-polar lipid. They require the carrier protein of the membrane to facilitate the transport across the membrane. A few ions or molecules are transported across the membrane against their concentration gradient from lower concentration to higher concentration. Such a transport is energy dependent process in which ATP is utilized and is called active transfer. Example, sodium potassium pump. Cell walls. क्या cell wall? As you may recall, a non-living rigid structure called cell wall. Non-living rigid structure, rigid, called cell wall, from an outer covering from the plasma membrane of the fungus and plant. Cell wall not only give the shape to the cell and protect the cell from mechanical damage and infection, it also help in cell to cell interaction and provide barrier to undesirable macromolecules. हाँ देखो, सेल वाल जो है ना सिर्फ शेप में काम नहीं आता, ये ऑल्सो टू द प्रोटेक्ट ऑफ द सेल फ्रॉम मैकेनिकल डैमेज एंड इन्फेक्शन और ऑल्सो सेल टू सेल इंट्रैक्शन एंड प्रोवाइड बैरियर टू द अनडिजायरेबल माइक्रो मोलिक्यूल्स. माइक्रो नहीं मैक्रो, बड़ा वाला. हाँ, एल्गी हैव सेल वाल मेड ऑफ सेल्यूलोज, ग्लैक्टोनस एंड मेनस. Cellulose, glutinous and mannose, and mineral like calcium carbonate, calcium carbonate. While in other planet it consists of cellulose, hemicellulose and pectin and protein. The cell wall of the young plant cell is called the primary cell wall. Is capable of growth. The cell wall of young plant is called primary wall. Is capable of growth. While gradually diminish, the cell mature and the secondary wall is formed on the inner toward membrane side of the cell. While gradually diminish as the cell mature and the secondary wall is formed inner toward the membrane side of the cell. Okay, the middle lamella is a layer mainly of calcium pectin. हाँ, मिडिल लेमिला जो है, मिडिल लेमिला इज ए लेयर mainly composed of cell calcium pectin which hold or glue two different neighborhood cells together. The cell wall and the middle lamella may be transferred by plasmodesmata which connect the cytoplasm of neighborhood cell. क्या बोला यहाँ पे? The cell wall and the middle lamella may be transversed by plasmodesmata, मतलब ट्रांसफर से जुटता है by plasmodesmata which connect the cytoplasm of the neighborhood cell. Okay, endoplasmic reticulum. While each of the membrane organelle is distinct in the term of its structure and function, many of these are considered together as endomembrane system because of their function are coordinate. The endomembrane system include endoplasmic reticulum, ER, Golgi complex, lysozyme and vacuoles. Yes, since the function of the mitochondria, chloroplast and peroxisome are not coordinate with the above compound, these are not considered as an endomembrane system. अब आते हैं endoplasmic reticulum. Electron microscope study of eukaryotic cell revealed that the presence of network or reticulate, network को ही हम लोग reticulate बोलते हैं, network or reticulate of the tiny tubular structure skirted into the cytoplasm. This is called the endoplasmic reticulum. It's called the endoplasmic reticulum. Hence, endoplasmic reticulum is divided into the intracellular space into the two distinct compartments, laminal मतलब inner inside endoplasmic reticulum and extralaminal मतलब cytoplasm compartment. यहाँ पे क्या बोला, इंडोप्लाज्मिक रेटिकुलम डिवाइड करता है कैसे, इंट्रा सेलुलर, इंटर नहीं, इंट्रा सेलुलर स्पेस इनटू डिस्टेंस कंपार्टमेंट, लेमुनल मतलब इनसाइड ईयर एंड एक्स्ट्रा लेमुनल मतलब साइटोप्लाज्म कंपार्टमेंट. ओके. The endoplasmic reticulum often show ribosome attached to the outer surface. This endoplasmic reticulum is bearing ribosome on their surface is called rough endoplasmic reticulum. In the absence of ribosome, they appear smooth and are called smooth endoplasmic reticulum. Endoplasmic reticulum, rough endoplasmic reticulum is frequently absorbed in the cell activity involved in the protein synthesis and secretion. प्रोटीन सिंथेसाइज में एंड सिक्रेशन में भी मदद करता. ओके. दे आर एक्सटेंशन एंड कंटिन्यूस विद द आउटर मेम्ब्रेन ऑफ द न्यूक्लियस. नहीं समझे. रफ इंडोप्लाज्मिक मेम्ब्रेन जो होता है ना, दे आर द एक्सटेंशन, क्या होता है, एक्सटेंसिव एंड कंटिन्यूस विद द आउटर मेम्ब्रेन ऑफ द न्यूक्लियस. आउटर मेम्ब्रेन ऑफ द न्यूक्लियस. हाँ. इंडोप्लाज्मिक रेटिकुलम जो होता है ना, एक्सटेंसिव एंड कंटिन्यूस होता है ऐसे. हाँ. विद द आउटर मेम्ब्रेन ऑफ द न्यूक्लियस. द स्मूथ इंडोप्लाज्मिक रेटिकुलम इज द मेजर साइट फॉर सिंथेसाइज ऑफ लिपिड. हाँ, मेजर साइट for the synthesis of lipid. In animal cell, lipids like steroid hormone are synthesized by smooth endoplasmic reticulum. हाँ. देखो, स्मूथ इंडोप्लाज्मिक रेटिकुलम, क्योंकि इस पे राइबोसोम नहीं, ये राइबोसोम बेरिंग कर रहा है. रफ इंडोप्लाज्मिक, न्यूक्लियस होता है, ये न्यूक्लियस है और ये न्यूक्लियर पोर है. ओके. ये क्या है, गोल्जी अपरेटस. सीधा देखो ऐसे ऐसे मुड़ा हुआ है, इसको गोल्जी अपरेटस. क्रिस्टेनिया, बड़ा जो अक्षर में लिखा होता है ना, वो क्रिस्टेनिया होता है. ओके. गोल्जी अपरेटस. गेमेलियो गोल्जी 1898 में, 1900 से 2 साल पहले, फर्स्ट एब्जॉर्ब डेंसली स्टेन्ड रेटिकुलर स्ट्रक्चर नियर द न्यूक्लियस. हाँ, फर्स्ट एब्जॉर्ब क्या था, डेंसली स्टेन्ड, कैसा होता है, डेंसली स्टेट रेटिकुलर स्ट्रक्चर नियर द न्यूक्लियस. दिस वेयर lateral named the Golgi bodies after him. They consist of many flat, disc-shaped sac or crystenia of the 0.5 to 1 micrometer in diameter. कितना, 0.5 micrometer to 1 micrometer. हाँ. They are stick parallel to each other. हाँ, कैसा होता है, they are stick, कैसा होता था गोल्जी बॉडी, stick parallel to each other. Okay. Varied number of crystenia are present in complex Golgi complex. हाँ, varied number of crystenia present in a Golgi complex. The Golgi crystenia are concentratedly arranged near the nucleus with distinct convex seams or forming face and concave trans or the maturing phase. The cis and trans phase of the organelle are entirely different but interconnected. The cis and trans phase of the organelle are entirely different but interconnected. The Golgi apparatus principally perform the function of packaging material. Golgi apparatus जो है principally perform करता है the function of packaging material का. Okay. To deliver either of the intracellular target or secretory outside the cell. हाँ, to be delivered either to the extracellular target or secrete out of the cell. हाँ. Golgi apparatus principally perform the function of packaging material to be delivered either to the extracellular target or secrete out of the cell. हाँ, यही काम करता है. हाँ, या तो इंट्रासेलुलर टारगेट पे सिक्रेट करेगा, आउटसाइड ऑफ द सेल. ओके. Material to be packed in the form of vessel. Form the ER fuse with the cis phase. इंडोप्लाज्मिक रेटिकुलम फ्यूज करता है कैसे, cis phase of the Golgi apparatus and move toward the mature phase. Move toward the mature phase. हाँ, Material to be packed in the form of vessel from the ER fused with the cis phase of the Golgi apparatus and move toward the mature phase. Move करता कैसे mature phase के. This explains why the Golgi apparatus remain in the close association with the endoplasmic reticulum. This explains why the Golgi apparatus remain in close associated with the endoplasmic reticulum. A number of proteins synthesized by ribosome on the endoplasmic reticulum are modified in the crystalia of the Golgi apparatus before they are released from its transphase. A number of proteins synthesized by ribosome. A number of proteins synthesized by ribosome on the endoplasmic reticulum are modified by the crystalia of the Golgi apparatus before they are released from its transphase. Okay. Golgi apparatus is the important site of the formation of glycoprotein or glycolipid. Yes. Lysozyme. These are membrane bonded vascular structure. कैसा structure है? Vascular structure formed by the process of packaging in the Golgi apparatus. The isolated lysozyme vessel has been found to be very rich in almost all type of hydrolytic enzyme. Hydrolysis, lipase, fructose, carbohydrate. Okay. Optimally activity at acidic pH. हाँ, लाइसोजाइम जो एक्टिवली एक्टिवेट होता है एसिडिक पीएच में. These enzymes are capable of digestion of carbohydrate, protein, lipids and nucleic acid. Nucleic acid भी. Oh वाह. Vacuoles. The vacuole is the membrane bounded space found in the cytoplasm. It contains water, sap, and excretory product and other material not useful for the cell. Vacuoles contain करता water or sap or excretory product and other material not useful for the cell. The vacuole is bounded by single membrane. It's called tonoplast. In plant cell, the vacuoles can occupy up to 90% of the volume of the cell. In plant, the tonoplast facilitates the transport of a number of ions and other material against a concentrate gradient into the vacuoles. Hence, their concentration is significantly higher in vacuoles than in the cytoplasm. Concentration higher कहाँ होता है vacuoles में than in the cytoplasm. In amoeba, the contractile vacuole. amoeba में क्या होता था contractile vacuole is important for the osmoregulation and excretion. In many cell as in protista, food vacuoles are formed by the inclusion of the food particle. In many cell as in protista, food vacuoles are formed by the inclusion of the food particle. Food vacuoles are formed by the inclusion of the food particle.